This is a recording of the 2025 Roscoe Ellis Jr. Lectureship in Soil Science, presented by Dr. Alexandra Kravchenko, a professor of soil science in the Department of Plant, Soil, and Microbial Sciences at Michigan State University.
Thank you very much, and for the nice introduction, and especially thank you for the invitation for selecting me to come and deliver this lecture. It's a great honor. So let me get started with my presentation. So the topic is soil pores as habitats for microorganisms. So why this is the topic of great interest to me? One big reason is because soil pores and microorganisms and their interrelationships, they are closely related to soil organic matter. And as we know, soil organic matter is the key to soil functioning. It's a key, it's what makes soil, as we know it. It is also a major player in global carbon balance. So the thing is that formation, storage, protection, everything about soil organic matter is enabled by soil microorganisms. And there is this very complex community of microorganisms inhabiting soils. And how they interact with soil organic matter, or what they do for it, depends on their on the composition. It depends on the composition of that community and on the functional capabilities of those microbes. That is what they can potentially do. But also their contributions very much depend on the environment, really microenvironment as far as microbes go, where they live, because those define what they actually can do. And so soil organic matter acts in this very multifaceted role, creating those soil microhabitats, being protected in them, being a resource for microorganisms, being their product. So it's all very closely interrelated. So then if we look of what are the components of these microenvironments that might be of importance for microbes, if we look at this in a kind of broad picture, the things that are important for them are the same things that are important to for organisms at a large scale, and for us as well. So what matters are the resources that are available in that environment. What matters are the physical characteristics of that environment. What matters are biological interactions taking place. So more specifically, looking at this big picture now, specifically for what matters for microbes, let's say if we think about the resources. Okay. Somehow it doesn't work as well as when we tried it. So maybe let me just use the mouse. So as far as the resources, if we just crudely look at the soil, we can think about it as, this dichotomy of parts of the soil that could be classified as very high resource places. That would be rhizosphere, that is soil next to the roots, drillosphere, soil next to earthworm burrows, trichosphere, soil next to decomposing residues. That would be very high resource environments as far as microbes go. The low environment is what would typically be the bulk soil that doesn't have any inputs of organic or nutrients from anywhere. Physical environments, what will matter for microbes, just a few things listed here. availability of oxygen, availability and consistency of water supply, pH, redox. As far as biological interactions, what would matter for microbes is whether the habitat where they are is accessible to predators, whether it's accessible to competitors, whether it's accessible to disease agents like viruses. So one thing that is common to all of these characteristics when it comes to soil and microbes is that all of them are extremely spatially variable at microscale. So let me just give you a couple of examples from both my group study and literature. So what you see on the picture is a soil aggregate about 5 millimeter in diameter. And what you see is X-ray computer tomography scan of that aggregate. So what my former student did, she cut, well, quite a few of these aggregates into tiny fragments, like less than 1 millimeter, some of them. And in each one of those fragments, she measured total carbon. So what you see here, these rows of dots are the data on individual pieces from a single aggregate, like the CT6 would be one aggregate. So one thing that really is striking to me is how extremely variable the total carbon is within just that 5 millimeter aggregate piece. So like if I pick this one here, an S9 aggregate, total carbon in those little fragments of it go anywhere from 0.5% to 1.3 times that. So thinking of carbon as a resource for microbes, that's huge variability of resources just within a single aggregate. So what you see here is an example of microscale variability in water distribution. So I will come back to this example a little later, but what we did to produce this image, we were adding liquids, different liquids to intact soils. And what you see is the result of, again, CT scanning of that soil, where the two colors represent those two liquids. So how we did that, we had these soil pores and we just uniformly added water on the top and let it redistribute through the soil sample. So we would expect, well, that water should be there uniformly filling the pores of certain sizes. Well, as you can see, it's nowhere near that. There are extremely high variability with some pores being completely filled with water. That would be the blue ones, and some are just air-filled, like these ones. So again, thinking from perspective of a microbe, there is this huge difference between living somewhere here, where you are basically in completely saturated water-filled pore, or living like a few hundred microns away from it, where you are in a dry land. Here is an example of pH variability that's from a work by colleagues in Germany. Here is the root of a plant grown in a rice box. And here is a map of pH next to this plant. And the scale bar represents the range of pH values. So just pointing to a couple of spots here, here is pH of 7. Right there, and here less than a couple of millimeters away, the pH is 5. So it's giant difference as far as soil characteristics going, happening within a few millimeter distance. And I can tell you more, it's probably like before that root moved there, like a day before, there was no this pH of 5 anywhere. So that pH environment, pH characteristic of the microenvironments for microbes, not only extremely spatially variable, it is also extremely temporally variable because of the plant root impacts. So you might be wondering, well, what's all this to do with pores and why the title of my presentation was soil pores? The thing is that soil pores is that those environments, they are the ones that define where microbes would live. They define where the roots and their resources would go. They define where soil, large soil organisms like earthworms would go. They define where water and gas fluxes will take place. So looking at microhabitats for soil organisms, to me, basically means looking at the pores, looking inside the pores. What you see here is sort of a schematic diagram that we thought about when thinking about the variety of soil pores and what the conditions might be inside them. On the x-axis is the diameter going from 0.1 microns to 1 millimeter here. So these ones, less than 1 micron in diameter, these would be the pores that would be too small for a regular microorganism to live in. But they would be an optimal place for protecting and storing that carbon, that soil organic matter. Pores of this size range, few microns in size, they are the ones where actually individual bacteria can get in, where individual hyphae can get in, some root hairs can get in, but still it's not something, because of space limitations, it's not something where you would have a lot of action taking place. So the pore size group that we think is optimal as far as microbial environment, this is where most of the carbon processing takes place, and that's supported by not just our, but work of others, is the pores in this few tens to a couple of hundred diameter range. So this is where water is available, but there is not too much of it. This is where oxygen is available, but again, it's not too high, not in a too high concentration. This is where there would be a lot of root inputs. This is where bacteria can not just live individually, but actually have enough space to build colonies. And this is where predators can, some predators, of course, especially microbial sized predators, can get there anyway, but bigger predators wouldn't have access there. So in the subsequent case studies that I will present, I will focus on these two pore size groups, this few micron size, which I call small pores, and a few tens of micron size, which I will call large pores. And in case I forget to mention it later, those orange and pink colors I will be using for small pores, and bluish colors would be what I will be using for to represent large pores. Okay, so well, so that's all kind of, you know, theoretical musing about what things might be like out there inside the soil microcosms. But how do we study them actually experimentally? What can we do? So two major routes. One would be a traditional destructive aggregate-based approach. The other would be less destructive pore-based approach. Not that it's completely non-destructive, but sort of more trying to get a view in a more natural conditions. So aggregate-based approach, the big plus is that it's easy. We procure the aggregates and we look at them. Don't need to have any expensive equipment for that. So there are several deficiencies to this method, aggregate-based method. One is the key assumption that underlines the use of aggregates to study poor characteristics in poor environments. So what the assumption is that in large aggregates, there is large pores. Small aggregates, that's what correspond to small pores. So while in general that should be true, that pore exclusion principle should work, but when it comes to actual measurements and actual ranges of pores that matter, these associations between aggregate size and what that we usually get and the pore sizes that we are interested in, they don't really hold very well. Here is just one example from a recent study that I found. I was hoping actually to find a positive example of this good relationship from looking for the studies. Well, what I found didn't seem to be very promising. What you see here on the y-axis of this table are the aggregate size groups, and on the x-axis are the pore sizes. And inside the table are the correlation coefficients correlating aggregates of particular size with pores of particular size. So what my expectations would be from this pore exclusion principle that we would see this red, and sorry, red means positive correlation. So I was expecting to see this nice red diagonal of positive correlations between large aggregates, large pores, small aggregates, small pores. The only thing that was consistent was that my expectation is this number here. But for the smaller aggregates and smaller pores, it actually was this totally unexplainable negative correlation. So basically, when we get the aggregates and assume that they represent pores of certain size classes, they probably are not representing them very well. But another problem with aggregate-based approach, and in that, if you think back to this, what I was talking about the characteristics of microhabitats that would matter for microbes, let's say specifically the resource availability. Well, once we sieve the soil and get those aggregates, there is no way to know where they come from, what type of resource environment they come from. So in essence, it's like when archaeologists look at their, digging site and they pluck an artifact out of its historic layer. And my understanding that after that, the artifact that just loses most of its historical archaeological value because no one knows where it came from. Same here in a way. Let's say this was my studied soil. I sieved it. I got these nice plump macro aggregates, and let's say I take this one in particular. And the thing is, I have no clue where it was. It could have been here next to the roots in the rhizosphere. And in that case, this aggregate will represent a high resource habitat, or it could be somewhere from here, from bulk soil. And in this case, it would represent a low resource habitat. So yeah, and of course, availability of resources is extremely important for microbial functioning and what that habitat means. So again, note that this approach is, would be totally make things totally impossible, but just it will not be very efficient because, for each aggregate, we would have to find out what exactly it is like resource-wise. So what I would like then now to tell you about is the few things that people and our group in particular does when it comes to pore-based approaches. So one way of doing those pore-focused studies is actually create artificial mixtures that where we have materials like this one here with dominance of large pores or materials with dominance of small pores. And then we subject these materials, we either inoculate them with something, we incubate them, we kind of see what microbes do when they are in these artificially extremely porous or artificially low small pore size environments. Of course, this also has lots of deficiencies. Yeah, and so yeah, working with artificial materials always does. Another approach, and the previous example I showed you of the aggregate and cutting it into tiny pieces, that's also something that can be done. You know, georeferenced cutting of the intact soil fractions and exploring microbial or other chemical or biochemical characteristics in those fractions, and then correlating them with the information, the X-ray CT information about soil pores that would be available for this, you know, aggregate or intact fragment of soil. Of course, also has a lot of downside. The third method is working with intact soil cores, and adding substrates, different things, carbon substrates, water substrates to the pores of specific sizes. This is the least destructive of these methods, and this is the one that it underlines the two case studies that I will talk about. So I'm going to talk about this one in a little more detail. Okay, so how does that work? So we take intact soil cores, and then we basically put them into pressure chambers and add to them water or the particular substrate with a schedule of metric potential changes in those water versus substrate additions, organized in such a way that the particular substrate, in theory, would be dragged into pores of certain size. So what you see here is, on the figure is the picture of the system that we have built in our lab. And what you see is just an example table from one of the experiments that we did with this system, where we were adding in the stepwise manner either water, or at certain metric potential, we were adding the substrate, glucose in this case. So when we wanted substrate to the glucose to go into this 4 to 10 micron sized pores, we added it at metric potential of about 30 kilopascals. And that's what I will be calling small pore treatments in my future examples. When we wanted that substrate to go into big pores, 30 to 150 micron pores, this is when we would add that substrate, the glucose, at 2 kilopascal metric potential. So that's the idea. So as I said, this is the least destructive of these different approaches. What is the downside? It's extreme variability. So that same variability that I was telling you about earlier, it really affects where water goes. So in theory, we would like to think, well, if we add water at 30 kilopascal, all of it should go into pores of this size range. Well, let me show you what actually happens. So what we did in one of the earlier studies applying this method is we actually wanted to see where what goes. So here we were adding radioactive glucose. So our intact cores, we scanned them okay? And what you see in blue on these pictures are our target pores as visualized from CT. So in these first two figures here, where glucose was adding to large pores, the blue are the large pores as how we see them from CT scan. In this bottom row, glucose was added to small pores, and those blue dots is where the small pores are. And I exaggerated them because if I didn't, you wouldn't be able to see them. So they are much smaller than the big dots I have. So this is where the pores, our target pores were. And this yellow background, yellow to brownish background, is where our glucose went. How could we tell? It's a radioactive glucose, so we could do this mapping of radioactivity. So what you can see in the large case, we do have high radioactivity, this brownish stuff, sort of around large pores, not just in them, but a lot just getting out of them and being around. What we see as far as small pores, we do have some association between the blue dots and brown part, but it's not very impressive. So they are getting somewhat in where we want to put them, but that is far from being ideal. We did another test of that, still thinking where things go when we add them. This is where we added two different solutions. So this is what that figure I showed you earlier come into play. We added two dopant solutions, and then we used X-ray CT scanning again at multiple energies to actually see where did they go. So here is that example figure, and now I can tell you that blue was barium chloride that went into large pores, and orange is potassium iodine that went into small pores. So what you see on the figure are these two squares, rectangles, I should say, the yellowish one and the blue ones. On this range of pore diameters, they represent our target pore sizes. And what the dots and their arrow bars, what they represent is analysis of actual images where they went. So as you can see, there is sort of matching. So we do have most yellow close to the target. We do have most blue close to the target, but again, there is tons and tons of variability. So now that I gave you all this disclaimer of how high variability in this method is, you might be wondering, well, why she's still talking about it? That should be forgotten and, thrown away. The thing is that it actually does work when it comes to a lot of microbial characteristics. So a number of studies has been conducted with adding substrates to these pores of different sizes and looking at what happens with CO2 emissions. There are people very consistently always were finding substantial differences in emissions. There were differences in presence of predators. There were all kinds of differences that people were finding. So what then I try to justify myself in using this method is, well, even if despite all this huge uncertainty in terms of where our substrates actually go, it still has a detectable effect on microbial activities, then, well, something is there. Let's keep using it. Okay, so now that I told you all about the method, let me move to my case studies. And here is the first one, which has been completed, and I have most information and results about it. So what the objective of the work was is not just to look at the CO2 emissions from pores of different sizes, but to actually look at the microbial communities inside those different pores of bacteria specifically. And in this work, my group collaborated with microbiology colleagues who did all the microbiology work. So where our soils come for this experiment, they come from this field study at KBS, and we took soil samples after about 11 years of soil being subjected to these contrasting plant systems. First one is the one that I will refer to as bare soil. This is where most of the time, except for last 2 years before our sampling, the system had no vegetation. They just keep it completely bare. Then there are two perennial vegetations, monoculture switchgrass and polyculture system restored prairie. Okay, so we took our intact soil cores, we scanned them, so we have our images. We added, this time we added 13C glucose, and the reason was that it enabled us, well, not our group per se, but our microbial collaborators to do stable isotope probing, which allowed them to see who actually ate, who consumed that glucose, who had or in a more broader sense, which organisms had the carbon of glucose origin in them. Okay, so when we set up the study, we hypothesized that we will be able to characterize 4 different microhabitats. So thinking about this high resource versus low resource 2 groups, and high resource, again, that would be rhizosphere and detritus, and low would be bulk soil. And then, so we can, using our CT imaging, we can see where the roots are, where rhizosphere is, or detritosphere, I should say, because those roots were dead. And the second tier of classification was this rough grouping of physical environments into large pores or small pores. So what the thinking was, what the hypothesis was, that when we look at large pores, or we add glucose to large pores in prairie and switchgrass, and those large pores are in the detritosphere, that those would be our high resource, high oxygen, environment. Small pores, we also expected that in the detritosphere of prairie and switchgrass, they would be representing this high resource small pore environment. Then in bare soil, we thought, well, large pores should be low resource environment. But as far as small pores, we thought that in prairie and switchgrass, bare soil, there will be our low environment small pore type of location. So we had it all kind of nicely balanced, but then of course came COVID, and part of our samples and all the lab closures and mess that came with it, part of our bare soil samples were lost, so I'm not going to be able to show you complete picture. Now let me, so that was sort of what we thought as far as which environments might be. So now let me tell you about the results and what we actually found. What I will be doing, just showing some snippets, because I wouldn't have time for all variety of characterizations that we did as far as the pore space and other things we measured. So as far as connectivity, so not just the size, but connectivity physical conductivity of the pore space and liquid conductivity, water conductivity of the pore space. That's what matters for resource travel and microbe travel. So what we found, and again, these that came from analysis of these samples where we added liquids to pores of different sizes. So what we found is that the liquid that is added to small pores, it is much more disconnected. There are much more of these disconnected clusters of that liquid as compared to when liquid is added to large pores, and that a liquid forms a much more connected space. So that's, again, just one characteristic of the physical space. The resource characterization. So as I mentioned, why we know where the resources are is because from our CT scan, we can visualize the roots, well, former roots, we can visualize particulate organic matter. And what you see here are just a couple of examples from our switchgrass cores and from our prairie cores. So the greenish stuff is particulate organic matter or remnants of roots and other residues. And brownish are the pores. So the big difference that is not just that I picked these particular cherry-picked these particular examples, but it's very consistent that in switchgrass soil, this monoculture switchgrass that's been there for 11 years, the pome is dominated by these old large roots. In prairie, it's like tons of these tiny pieces, root fragments of variety of sizes. Once we summarize the data from all our images, this is how it looks. So what you see here on the first graph is average size of pome fragments, and just consistent with that visual, much more, it's like twice plus size those pome fragments are in the switchgrass as compared to prairie. Number of pome fragments, prairie is way, way higher than switchgrass. So now, just a side note, so what does it tell us as compared to what are just regular measurements of particulate organic matter, like volume of pome or mass of pome measured in bulk wouldn't tell? Well, this is our pome volume, just all pome in those samples. What you can see, just as we would expect, there are much less of it in the bare soil, but the volumes, total volumes, are quite similar to each other in prairie and switchgrass. But if you think about it from microbes perspective, what is it to microbe, this total volume of the pome? It's how much resources it can get from it. That's what matters. So again, looking at the images, what we did, we sort of calculated what we call active detritosphere from the pome images. That is the, and here we picked somewhat based on literature, but somewhat arbitrary 300 micron distance from pome fragments. As what we would call active detritosphere. It's on the lower side of what people usually report in rhizosphere, detritosphere studies. So what you see here is what percent of the total soil volume would be what we call active detritosphere in each of these systems. So while in switchgrass, 50% of the soil volume was what would be active detritosphere. In prairie, it's like whole prairie is basically one big detritosphere, 80, I think it's like more like 85%. So basically 85% of soil in the prairie, the microbes there have very close, very direct access to resources that are coming from particulate organic matter. So even though in their volumes, the poems are reasonably similar to each other here, prairie and switchgrass, as far as what that means, as far as resource accessibility to availability to microbes, it's a big difference. So here goes this our theory that small pores in prairie and switchgrass bulk soils would be representations of the poor, resource poor environments. Here it goes, because there is really no bulk soil in those prairie samples. Yeah, 15% is sort of bulkish soil. Everything else is a high resource environment. Okay, so again, just some snippets of what our microbial colleagues, our colleagues, microbiologist colleagues, I should say, we're finding as far as microbial results. So what you see here are just examples of some richness and diversity indices for bacteria. And blue is large pores and small is, pink is small. And sort of the first impression you might get is that, yeah, pinks are above, more diverse than blues. And that, if you think about it, that makes a lot of sense. You know, small pores is just as I was showing you, they are much more disconnected both in the physical space and in that liquid distribution in them. So in the disconnected space, of course, there would be higher diversity of microorganisms. But if you start looking closer, you can see that actually that very strongly happens in the prairie soil, where there is really a big difference in diversity between large and small pores. In switchgrass soil, there is some tiny, really not that visible difference. And in the bare soil, it's actually reversed. So what is the key message that I took from this figure is that we really cannot just look at these soil microenvironments from one single perspective. We cannot just look at them as resources, and we cannot just look at them as physical space, because you know, small pores here in the bare soil, they're just as disconnected as small pores in prairie. The problem is that they do not have any resources. So it doesn't matter, you know, how complex that physical environment is. If there is no resources, no one is going to be there to actually create that diversity. And one side note, based on our other study that I want to mention, is that these differences in diversity, they can develop surprisingly quickly. Here we did a study where we created microcosms with pieces of plant residue in the center, and then these materials are dominated by either large or small pores, like in that first type of methods that I mentioned. And then again, my colleagues look at richness of bacteria and fungi organisms. They looked at them immediately on the surface of that leaf, and they looked at them at these two distances, at different distances from the leaf. And you can see that after just 24 days of that incubation, we do see this statistically significant difference with higher richness of bacteria, and also very surprisingly, even fungi developing in the small pore environments. So just some things that I found very fascinating when with my colleagues, we started looking at actual you know, classifications of microbes that only inhibited small pores versus large pores. So these are some of the organisms that were found only in large pores, never in small pores. So these three that tend to be plant polymer decomposers, these two that tend to be predators. And in small pores, these are the dissimilatory nitrate reduction capable organisms, were the ones that were found just in the small pores. And I'll return to this point in a bit. So even though overall there was a lot of overlap in terms of microorganisms, there are still, we were able, again, to my constant surprise, we still were able to detect those that were only present in one or not the other environment. And why they were there, plant polymer decomposes in large pores because they're root inputs, and predators because that's where that's where the prey is in large quantities. So keep me really excited and surprised that we're still finding something that makes sense despite all that huge variabilities that I mentioned. So what you see here are some more of that microbial results. These are top 10 in genera that were enriched in our carbon, the ones that we added as glucose. So the bottom brownish portions represent, so the width of these represents how enriched they were. And here is this long list of those top 10. So initially, at the 24 hours immediately after we added this glucose, basically, there were not that many differences as far as who was there and how abundant they were. Like that Pseudomonas, that purplish Pseudomonas was just everywhere. So like those same organisms jumped at the opportunity to have some free glucose in either large or small pores, either in switchgrass or in prairie. When things become more interesting is after 30 days of that incubation. So we are now not looking at microbes that ate glucose, because 30 days was ages ago and all that glucose long gone. We are looking at who was still utilizing that carbon. And there was this interesting difference in small pores between switchgrass and prairie systems, where we barely found those top 10 enriched organisms there, and the enrichment was very low. So what seems to happen is that those organisms living in the small pores of switchgrass, they were really not that good at benefiting from long-term, 30-day term, from that resource that they receive, unlike those living in prairie and unlike those living in large pores of both systems. So based on this and also some additional evidence that we just don't have time to present, our classification is now was adjusted like this. So in large pores, when we were adding our glucose to large pores, either in prairie or switchgrass, we were actually targeting this large pore high resource environments. When we were adding it to small pores, then in prairie, they represented small pore high resource environment. Again, that's your 85% of the pore, of the volume of the prairie soil being the detritus. But when we added them that to switchgrass, it actually represented small pores of the more on the side of the small pores that are low resource environment. So if we now think in general of what is happening with these microenvironments, the ones in the resource-rich large pores, this is where we would say the diversity of the organisms is relatively low. They are primarily plant residue decomposers, less so microbial residue decomposers. There are many predators, and there are copiotrophs as far as consumers. The resource-rich small pores, what we can say is that there is high diversity there, that residue decomposers would be, microbial residue decomposers would be more abundant than plant residue decomposers, and there still might be some predators, but there would be few. And in the resource poor small pores, at least in that version that we had in switchgrass, yeah, there would be no plant residue that composes there because there is no plant residues. There would be no predators, and this is where all kinds of oligotroph organisms we would expect to live. So you might be wondering, so what actually happens with the glucose that we were adding? And here are just one result for the 13C that we found in the soil in general. Well, immediately after 24 hours, we just added it. It was everywhere. There is no difference. But after 30 days, it actually became quite interesting, especially in the small pores of switchgrass. So while everywhere else it was just one low 13C still left, in switchgrass small pores, there was quite a lot of it left in the soil. And if you tie that to those 13C consumers for small pores and switchgrass that I showed a couple of slides back, so what thinking is that the organism didn't take it where it was. It remained in the soil. It wasn't, I don't have the results, that they were not in the dissolved form. The thinking is that most likely that glucose was attached to soil particles and probably conserved there and was protected, hopefully, in there. So the implications or partial conclusions from this case study, and actually my actual conclusions here, because the second case study I'll show you is still work in progress. So what our findings, what they mean for soil carbon processing. So these environments that we identified by looking at these specific systems suggest that in the large pore microenvironments, this is where the new carbon is added, and this is where it is actively consumed. And the chances for it to be protected or sequestered there are not that high. Most of it will be processed very quickly and gone as CO2. In the small pores that are rich in resources, this seems to be the best places as far as soil carbon, soil organic matter gains, because it is added there and it can be protected there. And the small pores in the resource poor environment is where protection potential is very high, because soil surfaces of soil minerals, they are there, they are empty, but carbon just is not coming there. Well, what we did in our work, we basically forced that glucose in there. This is not what happens in nature. So Nathan asked me a great question yesterday about, so what does this microscale research, how can it help us with, big practical questions? And so one lesson that I learned from these findings, especially regarding small pores that are resource poor, is that we really need to work with systems that are with the plant systems and soil systems, in order to minimize the volume of the soil that is not receiving the new carbon inputs. And that wouldn't just mean big carbon inputs, increasing the inputs themselves. That would mean to increase the spread of the input through the soil matrix. Specifically, that switchgrass, monoculture switchgrass that we used in this example, it is, as I'm sure most of you seen it and know it, it's this giant prairie grass with really massive root system. Yet it's again and again, everyone is finding that when it is grown in monoculture, it really doesn't increase soil carbon, as strange as it is. And the reason is just like what we are finding, is that there are these giant roots that keep reusing the same soil space and not putting out any carbon into the small pore environment. So in those switchgrass systems, the hypothesis, our next hypothesis goes, there is just a lot of soil matrix that does not have access to new carbon inputs, which it could in theory protect. So what would be a solution is to diversify those switchgrass, monoculture switchgrass stands by adding other plants that would increase root interactions and that would increase the volume of the soil that has this direct root inputs from direct carbon inputs from the roots. Okay, so some practical implications in terms of overall predictions of soil carbon processes. The thing is that it's, well, at least in theory, it's very easy for me to I give suggestions as far as modeling soil carbon processes while keeping in mind and accounting for those microenvironmental differences. Because using CT scanning, we actually can quantify all these things that define, at least roughly, the microenvironments. We know we can determine the volume of rhizosphere, volume of detritus. We can quantify the volumes of large versus small pores in either the detritus or rhizosphere, so we can characterize that. And then, knowing or assuming microenvironment specific rates of microbial activities, the modeling of soil carbon, soil microbial processes really can take into account this microscale things that taking place. So just a couple of words on the second case study that we are currently in the process of still wrapping up and still analyzing. So here we looked at carbon use efficiency. Roughly, that is what is the proportion of carbon that stays in microbial biomass and microbes, as opposed to the portion of carbon that is being lost as CO2. So that's a very important characteristics when we think about carbon protection and carbon storage. So just as in my first case study, we were looking at that by adding substrates to soils of contrasting textures. And now we looked at this different levels of nitrogen added together with the glucose. So we looked at the three soils from 3 sites. Basically, the reasoning was the study I just showed you, it's just one site, one soil. Could it be that what we are finding is just a specific case that only relevant to this particular soil? So the idea was to expand to soils of different textures and basically repeat, if not the same, but similar things. So that was the reasoning for looking at multiple soils. The reason for adding nitrogen was, thinking about things that affect carbon use efficiency. If we look at what happens in large versus small pores, who lives there, and what are microhabitat conditions, the expectation was that because we would expect small pores populated by oligotrophic organisms with low turnover, high diversity, low fungal to bacteria ratio, we would expect higher carbon use efficiency in small pores. But as far as environment characteristics, there is quite a lot of variations in what different components of environments in large versus small pores, what effect they could potentially have on carbon use efficiency. And specifically availability of nitrogen, we expected that there would be more of it in large pores than in the small ones, and that would have a positive effect on carbon use efficiency. So let's see, let me skip this because I think I'm running out of time. So by adding nitrogen together with our glucose, what we expected is that we will kind of balance, that is, we would increase that carbon use efficiency in small pores even more. So they would not be efficient just because of their microbial communities, but they would be even more efficient because they will get nitrogen. These are our results. And when we added glucose only, everything was nicely consistent with our hypothesis and expectations. So in all three soils, despite their big differences in texture and carbon levels, we consistently saw higher carbon use efficiency in small pores. But when we added nitrogen, then things didn't go at all consistent with our expectations. So like especially in our sandiest site, things went backwards. Our glucose with nitrogen, instead of increasing carbon use efficiency in small pores, it's actually decreased it. So I have no conclusions and no any ideas of what could be happening there. It could be that nitrogen addition actually changed microbial communities. We're now speculating very far and wide and what could happen, or maybe our hypothesis didn't make sense to begin with. But so that concludes my second case study. And I would like to acknowledge my group. This is my group members and their families at one of our lab parties, and many funding sources. And I'm very grateful for your attention, and see if I have time or any questions.
Thanks for the great presentation. I'm curious, how did you, in your C13 addition, glucose addition, you said that you had more C13 glucose in the small pores that was still there. But how do you know that it's glucose and not microbial necromass or
I
debris?
don't. So what was it? So I don't know. It's definitely not glucose. So sorry if I misspoke and said that that's glucose. It is student C of carbon of glucose origin. So that's what was long time ago added as glucose. So besides looking at the total soil certainty, we also looked at microbial certainty, and there were no differences in microbial certainty. So it wasn't really in the microbes, it wasn't in the dissolved carbon, it was in just this total soil carbon.
But could it have been dead microbes that had?
It could be dead microbes, yes, either dead microbes or either necromass or what just attached to the soil. Yeah, either one of those. That's very true.
And then your last point there at your last slide about carbon use efficiency, I guess I would have predicted that nitrogen would decrease carbon use efficiency because you'd be narrowing the CN ratio and favoring respiration rather than maintaining or retention of carbon.
Yeah, that's probably what we would need to think about, and from that perspective, because thinking from literature, what people keep saying is that whenever there are stresses and microbes have to spend carbon on something to overcome those stresses. This is when carbon use efficiency decreases, right? So then that's what our thinking with nitrogen, that if they are stressed for nitrogen and we supply it, then they should be, yeah, they should feel good and build themselves up. But yeah, I guess we need to do a lot of second thinking about what exactly could be happening.
Sasha, thank you for a nice presentation. What do you think about the mineral contribution? Like so you look at the physical, chemical, likewise, and then the pore system, smaller pores versus larger pores. And then you could have, when you're thinking about those pores, you could have in real soils, naturally, you could have different mineral types with different characteristics.
Yeah, this is great question. Indeed, this is what partially we try to get at with the second study, looking at wider range. But our range, even though we did have these differences in texture and overall total carbon, mineral-wise, those were same glacial deposits that one couldn't really tell apart that much. So they behaved quite similar. So what, and again, we haven't really looked ourselves at drastically different soils with drastically different mineralogy. But from what I've seen in literature, like recently there's been a paper by a Kravchenko group in SBB where they looked at carbon use efficiency in, I think, is 5 or 6 soils from Europe, representing quite a range of minerals, and definitely different from what we had. Some of them were. So they reported quite similar differences in terms of how CO2 is emitted when substrate is added to large versus small pores, and what happens with carbon use efficiency in large versus small pores. So yeah, we haven't really looked at tropical soils, and that is where things might be very different, but that's next on the list.
So what inspired you to become a soil scientist?
Well, I wanted to be a biologist, and soil science seemed to be very close to it. Yeah, but well, maybe, I don't know, maybe my grandmother, because she was a researcher studying sugar beets. So she was sugar beet breeder. And, you know, yeah, we lived together with my grandparents, and the house was filled with all kinds of papers on sugar beet breeding and her graduate students all over the place. So I thought, yeah, that biology and soils and agriculture are cool things from very early age.